Good morning, everybody. As always, thank you for being with us this morning. We're just giving it a moment for everybody to join into the webinar. We have our own Dr. Euan Ashley presenting today. It's a talk that I've been looking forward to for months, so I can't wait to hear Dr. Ashley speak. I'll turn it over next to Dr. Harmon. Dr. Harmon is going to talk about our upcoming Women in Medicine photo. Steph, I believe we've been doing this now since 2019. We did it through the pandemic. We did a mixed versions online and in person, and we have another one coming up, right?
Yes, in anticipation of Women in Medicine Month in September. You saw an announcement go out in our weekly newsletter for the Women in Medicine photo, which is now an annual event. And as similar to last year, we are doing a hybrid approach where we'll have an in-person photo on the 14th, Wednesday, noon at the Clark Center steps. Wear red, your white coats, bring your masks outside for the photo. And then the virtual photo will take place the same day at 4 p.m. And you should have received the Zoom links for that in your emails. So looking forward to seeing everyone virtually and in person. Thanks.
Thank you, Dr. Harmon, for leading this effort and for all the team behind the scenes also helping with logistics. So looking forward to seeing everybody. Wonderful. And I just want to briefly mention our upcoming grand rounds. We have next week a couple of our own speaking regarding the Diversity Inclusion Awards. The winners of the grants, Drs. Young and Clark, will be presenting on the work that they're doing. So we're really looking forward to seeing them speaking then. So please join us for that. And that being said, now we're excited for our main event. I want to turn it over to Dr. Lewis to introduce Dr. Ashley. Dr. Lewis, thanks so much for being with us today.
Thank you so much, and it's really a pleasure to have an opportunity to introduce Dr. Euan Ashley. He's a native of Scotland and a graduate from the University of Glasgow in 1996 with degrees in physiology and medicine. Afterwards, he completed his medical residency, and he also got a PhD in 2002 in molecular physiology at the University of Oxford. Afterwards, he came to Stanford University, where he trained in cardiology and learned how to be an outstanding CCU doctor under Dr. Randy Vagelos, and joined the faculty in 2006, where he regularly attends in the CCU and on the heart failure service, and also sees patients in the ambulatory setting in the rare diseases clinic. In 2009, he actually founded the Stanford Center for Inherited Cardiovascular Disease, and it's now become one of the largest integrated care of patients with inherited cardiovascular disease in the world. It's really epitomizes how we should manage patients and really leverage our understandings of genomics to provide precision health. He's the Associate Dean at the School of Medicine for Precision Health, and last year he became the inaugural Roger and Joelle Burnell Professor of Medicine and Genetics, and this recognizes his contributions to cardiovascular medicine and genetics. He's the author of almost 400 publications. He has an incredibly large and talented research group focused on the science of precision medicine, data science, artificial intelligence, and digital health. He's the PI of multiple grants, but including My Heart Counts Cardiovascular Health Study, which was launched in collaboration with Apple. He's a co-investigator for the AHA One Brave Idea with Callum McRae. It's a $75 million funded project, and various projects, including the Precision Medicine Initiative for All of Us. He also led the team that carried out the first clinical interpretation of the human genome and established this sequence as now a routine part of the care of patients with genetic disease. And we can now take this for granted. He's built a rapid turnaround computational pipeline for sequence interpretation and made the first clinical diagnosis via long-read sequencing. And in fact, is the Guinness Book of World Records title holder for the fastest DNA sequence technique. Last year, he published his first book entitled The Genome Odyssey, Medical Mysteries and the Incredible Quest to Solve Them. And he's the recipient of multiple awards, including the AHA's Medal of Honor for Precision Medicine in 2018 and the National Innovation Award in 2009. He's co-founded 4 biotech companies in diagnostics, therapeutics, and analytics, and is just a well-rounded person. He's an amazing clinician, a scientist, and most importantly, an amazing person. So it's really a pleasure to introduce Dr. Euan Ashley, and he will talk to us about the need for speed, ultra-rapid genome sequencing for critical care. Dr. Ashley.
Well, thank you so much, Eldrin and Errol, and thank you for the invitation to join today. Well, let's just get the slide going. I'm going to talk about the need for speed, and specifically the need for speed in genome sequencing for patients in critical care. Before we get moving, here are my financial disclosures. You heard a little bit already from Eldrin about some of the companies that we founded from technology developed in the lab. Not that long ago, Nature suggested that as well as financial disclosures, academics were potentially influenced by other things, such as institutional loyalty, personal beliefs, and ambition. So I've actually started including this slide because I kind of believe that these are probably more important than financial disclosures in some ways, but I leave them for you to read. In particular, I have a love of technology, and so being in Silicon Valley, this is a good place to pursue that. Well, it's not a surprise to anyone in this audience that inside every single one of us, inside almost all of the cells in our body, is a code of 6 billion data points, 3 billion bases, right-handed double helix DNA tightly coiled inside 23 pairs of chromosomes. If you take that DNA out of every cell in your body and stretch it end to end, it would reach to the moon and back thousands of times. It's a lot of information. In fact, if you were to stare at your genome at this page as it scrolled by, you would have to wait 3 months watching 24 hours a day, 7 days a week before you saw your entire genome scroll by. That is more data than we're used to dealing with in medicine. Where the story begins today, though, is June 27th, 2000, where the genetic code of human life is cracked by scientists. At least if the New York Times said it, must be true. We have a little update to some of that we'll come to a little bit later. But here, of course, is a younger looking Francis Collins. Craig Venter, Bill Clinton was off to the side. And this was the initial release of the draft sequence of the human genome. It really, in the end, was actually more like 92% of half a genome, but we'll come to that later. This was a major achievement. 10 countries, 10 years, $3 billion of funding. But if a genome still costs $3 billion, we wouldn't be here talking today about its impact on medicine. The next genome was that from Craig Venter, and it's traditional to talk about how quickly genome sequencing has dropped in price. So let me make sure I do that at the beginning of this talk. The next genome was $100 million. That's Craig Venter's genome. And over the course of the next 8 years, genome sequencing dropped in price so much that it was compared to Moore's law, not just in Silicon Valley, where we are often talking about the speed that technology moves forward. Gordon Moore, along with Bob Noyce, was one of the co-founders of Intel, and they defined this idea that every year to 2 years, there would be a doubling of the number of transistors in an integrated circuit, which would lead to these leap forwards in computer technology that we're all very familiar with. And of course, over the first 7 or 8 years, as you can see in this graph, the drop in genome sequencing tracked Moore's law, and that was pretty amazing. But in 2008, something remarkable happened. The cost in genome sequencing dropped at such a rate that it left Moore's law in the dirt. In fact, we got to the point where you could sequence a human genome for around $1,000. And in fact, that has dropped further. I've shown this graph, many have shown this graph over many years, and it is spectacular. But I found a few years back that a slightly more tangible way of demonstrating this. I used to drive past the Ferrari and Maserati garage on El Camino Real on my way home to my home in Menlo Park, and I would stare at this car slightly out of my price range at $400,000. If this car dropped in price as much as human genome sequencing had dropped in price, at the time the stories I'm going to tell you today began, it would cost as little as 40 cents. Over the course of the next few years, through the stories I'll tell you in the next few minutes, it dropped to a $1,000 genome, a 10-cent Ferrari. At the moment at Stanford, the reagent cost for a genome is around $500, a 5-cent Ferrari. And this fall, several companies will release a $100 genome. A $100 genome is a 1-cent Ferrari. This is an incredible, unprecedented change. And it's up to us to think about what is the impact that having this technology available can make for our patients, because there isn't a disease without some genetic component to it. In many diseases, it is not the most important component. In many diseases, it is everything. So our stories really begin in 2008. And in 2008, this gentleman was not someone I knew very well. He's now a good friend and colleague, Steve Quake. He is the president of the Chan Zuckerberg BioHub, now actually runs science for that institution. just as of the last month. At the time, he was chair of the bioengineering department at Stanford, and he had made headlines that summer for sequencing his own genome in a matter of weeks with 2 people in his lab for $50,000. Now, I've just told you, you will shortly be able to get a $100 genome, so 50,000 sounds pretty expensive. But to put this in context, the genome before his, the 4th genome that was ever sequenced in the world, cost $200,000 and took dozens of people months to put together. So to sequence a genome, including especially your own, in 5 days with 2 people in your lab, using a technology you invented, was something quite remarkable. And I was visiting Steve, actually, just to talk about something completely different, finding his office in the Clark building. And he started showing me his genome on the screen. So at this time we were running the clinic. Eldrin mentioned our Center for Inherited Cardiovascular Disease here at Stanford. We were sending gene panels, 8 genes. Would take 3 months, cost $5,000. And suddenly I was seeing a whole genome. Actually, there were only 5 people in the world who'd sequenced their genome at this point, and I was seeing one of them on the screen, not far from where we all practice medicine and do our science. It was a jaw-dropping moment. But then he started pointing out variants he had in certain genes, and some of them were genes I recognized, like myosin binding protein C. That is a gene in which variants cause hypertrophic cardiomyopathy. And he said, have you ever heard of that gene? You ever heard of that variant? And I didn't recognize the specific variant, but I certainly knew the gene. And so slowly, as he pointed out these variants that were potentially causing of cardiovascular disease, the scientific meeting turned into a medical meeting, and I started asking him about his family history. Like most people, he didn't remember it much at first, but over time, we worked out that he had a pretty significant cardiovascular family history. In particular, his father had a rhythm. He couldn't quite remember the name of it, ventricular tachy something. Well, it turns out his dad had VT, there's ischemic cardiomyopathy, coronary artery disease, aneurysms, and indeed in his cousin's son, a 19-year-old black belt in martial arts with no previous history of any medical condition, died suddenly aged 19. And so that meeting changed from a scientific meeting into a kind of medical meeting. And as I invited him to our clinic to be checked out for the significant family history, we also realized he was about to become the first patient in the world to walk into a clinic with his genome and his genome on a hard drive. And I think we realized at the time that really nobody had yet worked out what to do with that. It seemed like it would be relevant. We knew from twin studies that 50% of the heritability of cardiovascular disease, that there was a 50% heritability of cardiovascular disease. So the genetic component, pretty significant there. Now we had a whole genome, what were we going to do with it? Well, how do you analyze a genome? Well, the first thing you do is you take a blood sample. You take a blood sample in order to get the DNA from the white blood cells. You then blast it with ultrasound, essentially to break the DNA up into fragments, and you feed those fragments into one of a few pretty dominant machines from a few different companies, and we'll talk a little bit about some technology over the next little while. And then you end up with a text file with a bunch of reads, basically letters, A, C, Gs, and Ts. For the Illumina platform, that is the dominant one, these tend to be 150 base pairs long. So you have a file with millions and millions of rows, and in each one of them, there is 150 base pair read. So then you basically take the human genome reference, the one from the Human Genome Project, and you do a process called alignment, which is to say you take each one of those reads and you find the place where it matches best. The algorithm for using this is actually from compression technology. It's called Burgess-Wheeler. And then once you have that, and this is quite literally called a pileup, because it's a pileup of reads, you then send another algorithm along to basically read the letter or 2 letters, depending on the position of each of the place for the genome of interest compared to the genome of reference. In this example here, you can see the patient would appear to be homozygous for A, whereas the reference is T. Important note, the reference is monoploid. It's only one. It's not a diploid genome. There's only one series of letters. But that is important for a number of things, including for some of the next generation discoveries that are coming probably in the next few years. So that's what you do, and then you end up with a variant call file. And in some ways, that's where the work really begins, because usually a variant call file has 3 or 4 million variants, and you have to decide now which ones are important, which ones are not, which ones are relevant, and that's a mechanism of filtering predominantly. Of course, we remember that there's a 3-letter amino acid code. Here's a good example. If this is the reference, series of words that you recognize. If we make a substitution in that, we change the meaning, of course. And so a single nucleotide variant change will change the meaning, but will not drastically change the reading frame of the gene. Of course, if you insert or delete a letter, you drastically change the reading frame of the gene. And so those are much more destructive variants. You can also introduce a start that wasn't there before, or a stop that wasn't there before, or change the splicing for intron and exon boundaries. So a number of ways that variants can alter the 3-letter amino acid code. At the time we started this with Steve Quick, there really was no way of doing this on a genome-wide basis. Of course, the basic science was known. And so we turned to the amazing campus we have here in Stanford, and I basically asked a bunch of friends if they'd be willing to join in this idea, slightly crazy idea at the time, of trying to analyze one genome in the context of a single person, but also in the context of everything that was known at the time about medical genetics. And I went to a close friend and colleague at the time who looks just a little bit older than he did back then, Matthew Wheeler, now one of our assistant professors, of course, in cardiovascular medicine. And I remember we sketched out this on a whiteboard to try and decide how would you start to think about destructive variants in the genome across an entire genome in a single individual, in an individual with this kind of family history. We worked with others. Atul Butte's group was here at the time, and 3 individuals from his group shown here all contributed to thinking, if you had this information available, how would you bring it to bear on a medical visit? And in particular, how might you bring it to bear in a visit where you were thinking about preventive care? So we came up with ideas like this, where your pre-probability is demonstrated here according to your age and size and weight and a number of other demographic features. And then we layer on top the addition or subtraction of your genomic risk. It was kind of futuristic. We didn't really have the population data to support it at the time, but it was kind of an imagination kind of exercise. What would it look like if everyone had their genomes available? And Joelle Dudley in particular came up with this kind of graph where the font size of the individual risk factors would vary according to the ultimate risk that you had, and the individual arrows would point to the relationships we knew between environment and gene, because everything at the end of the day is going to be a result of the interaction between lifestyle, your environment, and your genetic predisposition. So this is ancient history. I'm not going to spend too long for this, but it was something that we were surprised with when we were excited by this study, but surprised even at the amount of interest in it. I think there was some kind of thought that maybe personalized medicine was within reach, that this was something that we could reach for, the idea that maybe you don't give the same drug to every patient. Maybe there's a way of thinking about prescribing that is more personalized, a way of thinking about risk factors that is more personalized. And as Eldred mentioned, we had the privilege of working with the Obama administration. There was even for some time a little exposition at the Smithsonian on this work. But this was just the beginning for us. This is 2010. This paper was published. Of course, we were interested not just in people who had risk factors, but in patients. And in particular, we were interested in some patients in whom diagnosis would need to be made pretty fast. And an example of that is a patient who was referred to us when she was still in gestation, 36 weeks of gestation. It was found that she had a heart rate of 70 beats per minute, which you will recognize as about half what it should be for a little baby swimming around in her mother's womb. She was transferred to Stanford Children's Hospital, delivered by cesarean section. And at that point, it became clear under the care of Ann Dubin that she was suffering from a very severe form of long QT syndrome. So heart block was on our differential because of the low heart rate, but in reality, this QTC corrected at 610 milliseconds was long enough that it was blocking the next beat. And as you know, a low heart rate isn't really a problem for a compliant newborn's heart, but torsade de pointes is absolutely a problem as a potentially fatal heart rhythm, turning of the points shown here. And on her first day of life in the outside world, this little one had 5 cardiac arrests, and 5 times the team was called to the bedside for CPR. Some of these were self-limiting, some required CPR. But we know also that she became the youngest baby ever to have a defibrillator implanted. Just a day, I think, later, she had this defibrillator implanted, as you can see in her abdomen. Obviously, no room under the clavicle as we do for adults, and this was implanted in the abdomen, and the lead going up from below towards the heart. We know that long QT syndrome is a genetic syndrome. We also know that there are 3 common versions, 2 of which hit potassium channel genes, 1 of which hits sodium channel genes. We have medications that we can target towards those. We have sodium channel inhibitors, particularly like mesillatine, and we know that beta blockers are particularly potent for potassium channel long QT syndrome. So if we were able to tell what the genetic basis of this was quickly enough, we could really potentially help and do precision medicine, do precision prescribing for this little one. The problem is, at the time, a genome would still take 3 months if you ordered it, and it would cost many thousands of dollars. But we'd heard of a new way, and working with Illumina, it was possible to put aside a whole machine just for one genome. And for a little one in the neonatal intensive care unit, it seemed like there was probably no better reason to do that. So we called some friends at Illumina, and using a technique developed along with Stephen Kingsmore, who at the time was in Kansas, we sent the blood basically in this express envelope. And this is in fact the result coming back. We sent the blood on a Friday, and the result was back just a few days later, Monday or Tuesday of the next week. Over the weekend, a very talented at the time postdoc and recent faculty member here in Stanford Children's Health and Cardiovascular basically speeded up our computational pipeline. This is an evolution of the pipeline I told you about a few minutes ago, first developed by Matt Wheeler and developed by others in the lab over the time. He managed to knock about 10 or 20% off this time over the weekend while we were waiting for this genome to come back. And so the minute the data came back in that envelope I just showed you, he ran the algorithms, and we basically discovered 2 variants of interest. One in the potassium channel gene I mentioned earlier. This is the hERG channel, KCNH2, long QT2. This one inherited from the father. That'll become important in a moment. And this gene that's well known from genome-wide association studies of long QT hadn't been implicated in long QT in a Mendelian manner, but this is a frame shift. So this is a very disruptive variant that we were pretty certain contributed. Over the course actually of many years, this was followed up. This work was published in 2014. One of our collaborators, Nipavan Chiamvimunat at University of California, Davis, followed up these specific variants to demonstrate our suspicion was indeed, in fact, true. This is work from whole cell patch clamping of the potassium currents. And just to quickly take you through it, she modeled the exact changes in this little one. Here's the wild type hERG current, the RNF207, and black is the control here. And this is the patch clamp shown on the right. If you take just the hERG variant here, but the wild type 207, you can see in red that in fact, it's pretty maintained, the potassium current. But if you have both together, which is the case with this little baby, each individual parent had one of these, but the baby was the only one with both, you can see a very significant drop in the potassium channel late tail current here, really demonstrating, one, that it takes a long time to do the basic science workup, but 2, thankfully, after all these years, we were right in targeting that this was a potassium channel gene. And so we were able almost immediately to withdraw the rather toxic and minimal therapeutic window sodium channel mesillatine doses to really focus on what was working, which were the beta blockers, and managed to get her out of the hospital. This was a picture of her just 6 months later, and I actually have an updated picture now from 5 years later, which is where we're at with her most recent birthday party. She has not been back in hospital, is actually doing really well, and I think still fairly unaware of the role that technology played in the early hours and days of her life. Well, sometimes the diagnosis is clear and we do an ECG and it's the longest QT we've ever seen and we know what it is, we just don't know the mechanism. There's other times when we have no idea what's going on, and for patients and families who are suffering undiagnosed disease, this is a long journey. It's a long journey of going from doctor to doctor to doctor with nobody really having answers, nobody having a label, nobody telling you anything about it. And meanwhile, if you're the individual seeing your functions decline, if you're a parent, standing by helpless as your kids appear to be deteriorating with no answers. There's a reason that this is now called a medical odyssey, of course, from Homer's The Odyssey. Odysseus was the king of Ithaca, who spent 20 years trying to get back to his betrothed, fighting multiple shipwrecks, one-eyed, crazed, man-eating giants, the hypnotic siren songs of a beautiful woman, and crazy monsters with dog heads and shark's teeth. There's a reason, I think, that we call this a medical odyssey. And so we think about patients as going through that kind of journey. This, of course, is the Odyssey. If you're interested in having a read, it's a pretty long read, I would say. I noted when I found this on Amazon that it was called one of the 10 best books of 1996, which I thought was quite interesting. Also on Amazon, if you're interested, you can actually follow the author, just in case Homer decides to write any more epic books. But the Undiagnosed Diseases Network was what was put together and pulled together initially by the NIH to try to answer these kind of medical odysseys. There had been a center at the NIH itself, at the medical center there for a few years, started in around 2009 by Bill Gall. But the idea was spreading that. It was being able to do this regionally, being able to do it for many more patients, and thinking about the real excitement that the genome brought to being able to solve rare and difficult medical disease. And we were very privileged to be part of it. We're still part of it now, although it's in a transition phase after its first 10 years of funding from the NIH. But we're 12 clinical sites. There's a coordinating center. There's medical sequencing. Almost everyone gets the genome sequence. Metabolomics core. Also, we're able to do model organisms, rapid turnaround, trying to work out the answer when you get a particular gene list from a genome study. And one of the things that I have really loved about the Undiagnosed Diseases Network is that the first thing we do is traditional medicine. It's all about human interaction. It's all about meeting the families, talking about their stories, trying to pick up clues. It's almost like the Sherlock Holmes approach that many of us thought we were going to be doing all the time in medicine, but happens not quite so often. But it's human interaction, it's detailed history, it's traditional tools of medicine, and also finding the right expert wherever they are in the world. But one of the joys of doing that here in Silicon Valley is that we can also combine that with cutting edge technology. So almost every patient has genome sequencing, often multiple members of the family do. We have metabolomics, model organisms, iPSCs, and immune repertoire sequencing on hand, basically to try to solve these most difficult of cases. And I'm going to give you one example. This is one shown here. These are two little boys, Carson and Chase Miller. Carson was the older boy, and he had been developing normally in every single way until about his first birthday, when he started to slip back on his milestones. He'd been cruising around furniture, he'd been feeding himself, and slowly over the course of a few months, he started to not be able to do those things. He spoke a lot less. His intellect appeared to be intact, but his motor functions were declining. And his parents, Danny and Nikki, took him to multiple doctors to try to find the answer. They were given the label cerebral palsy, but that really doesn't mean anything except brain paralysis, and certainly doesn't tell you anything about what's going on. Danny would spend hours at night coming home from work, Nikki having spent her days looking after their little boy, and he would go onto the internet and try to find answers. When the younger brother, Chase, was born, and he started to go through exactly the same thing, the years went by where this pattern continued. And Chase, first of all, had normal milestones, and in the same way that his older brother did, he started to slip back, couldn't feed himself, didn't speak, wasn't able to walk anymore. And just think about the torture that these parents would be going through as they watched their two beautiful boys start to slip backwards with having no idea what was wrong with them, having no doctor they could find with any idea what was going on with them, and not knowing where this would all end up. That's the medical odyssey that so many of our patients and families deal with. They've been, I think, to 5 neurologists. They've been to developmental specialists, 3 or 4 pediatricians. When they finally heard of the Undiagnosed Diseases Network, and living here in the Bay Area, they were referred to our center at Stanford. We sequence the entire family, which is what we do. And sometimes the answer just falls out. And in fact, just before I'll tell you that, sorry, here's the imaging. Basically, the MRIs of their brains showed basal ganglia lesions, but again, nothing that really pointed to a known diagnosis. Other extensive testing from all of these doctors was negative. So we sequenced their genomes, and in this case, the answer came out. And this is the actual pileup. I mentioned the pileups earlier. So these are the actual pileups lined up for the 4 individuals, the 2 siblings and the mother and father. And you can see that we have a case of compound heterozygosity, which is to say that a variant from each parent, just like in the long QT case, was given to each sibling. One variant on it by itself, not enough. Both mother and father were normal, but together in this unusual gene, mitochondrial trans-2-adenyl-CoA reductase. So the mother had an insertion, Nikki had an insertion in this gene, and this disrupted this in both boys. And then in the father, Danny had a 5' UTR, the untranslated region, had a splice change there that made a significant difference. So both copies were hit for both these little boys explaining their condition. This was a condition called Mepan syndrome that had been described only in 7 individuals to that point. So they were the 8th and 9th boys to be diagnosed with this condition. Now, I will forgive you for not being that familiar with the mitochondrial FAST2 fatty acid synthesis pathway. I don't even remember being taught this pathway. Certainly was not familiar with it, or remember that specifically that there was a fatty acid synthesis pathway in mitochondria, but believe me, there is. Starts with malonate, goes through this obviously complicated pathway. The MECR gene that I just showed you that was dysfunctional in these 2 boys is shown right here. And the really important part of this pathway is shown down here, which is what you get at the end of this complicated biochemical pathway. And in particular, one of the effectors of this pathway is lipoic acid. And I'll remind you that Danny had now spent years, 4 or 5 years, every single night looking on the internet, convinced that somewhere was the answer to what was ailing his boys and the answer to what would happen next. He did not realize, I think, that if we were able to give him the answer, which he did, then he could go and start searching for this gene, start searching for the thing that was missing, and thinking that while we can't replace the gene, perhaps we can actually replace what is missing. This lipoic acid is not some incredibly expensive gene therapy that you might have heard of, but rather something you could buy on Amazon. And so imagine his surprise when he realized he could buy a bottle of something that could help his kids for $16.10 and have it delivered the next day in an Amazon box. With the permission of his doctors, he did exactly that, started supplementing with alpha lipoic acid and a couple of other supplements under the care of the doctors here at Stanford. And over the course of the next few months, their neurological condition significantly improved. This was not a miracle cure. We're still hopeful for that. We're all still working on this, but a significant improvement in their function and reattainment of some of their milestones was something that he was incredibly happy to be able to witness and gave them some extra hope for the future. Over a 20-month period, we had 1,500 patients sent to the network, 600 accepted. We gained a 35% diagnosis rate. Remember, this is after they had been to dozens of doctors beforehand. 75% of the time that was through the genome, and 80% of those patients, there was an actionable change that could be made. And maybe more importantly, here's a new technology. The costs were 6% or prior. If you bring the genome earlier in these medical odysseys, of course, you're going to save money. And in the case that we modeled in this paper, the costs were 95% lower. We also have the genome available. Eldrin mentioned this, it's now routine. You can order this at Stanford, and we became the first center really in the world to offer routine genome sequencing, both if you order an exome for genetic disease, but now also a cardiovascular panel. So if you believe someone has an inherited cardiovascular disease, you will order a panel, but in fact, what you'll get is the whole genome backbone. This also means you can get polygenic risk scores in time, and pharmacogenomics, something we hope to add in the very near future. Polygenic risk scores are actually featured in this recent scientific statement. Jack O'Sullivan, one of our cardiology fellows, and chaired this scientific statement while he was still a resident here. So I wanted to highlight this, just came out a few weeks ago, really giving guidance to the community on when polygenic risk scores might be most useful for coronary artery disease and a number of other conditions. And you can see this pattern here where standard risk factors are not that predictive. Clinical risk factors and risk factors with polygenic risk scores are significantly more predictive. All right. Well, we've thought about cost and we've thought about accuracy a lot, but what I said I would tell you about today is speed. And so some patients, speed really matters. I gave you the example already of the little one. Here's another patient for whom speed really mattered. This is Matthew Kunzman, and he presented to his local hospital in Oregon with a cough in the first winter of COVID, and he was 13 years old. His parents and the doctors initially thought it was likely to be COVID because, of course, but actually after he had an echo, it was pretty clear this was not COVID, but rather dilated cardiomyopathy. And you can see that his heart was really not moving well at all. He had significant mitral regurgitation shown with his echo at the bottom there. He was essentially heading towards cardiogenic shock. So he was airlifted to Stanford Children's, where he rapidly deteriorated. He was crashed onto ECMO, and then shortly after underwent implantation of a HeartMate 3 device. This is it shown here. It's in his chest, and this is him looking rather pale but somewhat stable with the battery pack on his shoulder. The question, though, is what would happen next, because possible causes for a rapid decline like this include myocarditis, and genetic heart disease, myocarditis, potentially reversible. And in fact, there are plenty of stories of being able to explant these devices. But if he has a genetic heart disease, clearly that is not going to be reversible. And so the question is, how do we make that decision? Certainly we'll do a biopsy. In fact, he had one, although this is a left ventricular biopsy shown here. We normally do right ventricular biopsies. He did have one. It certainly showed the non-specific findings that we always find. The problem is that myocarditis is patchy and it doesn't really particularly well rule that out. So could we alternatively rule in a genetic cause by genome sequencing? Well, we're back in the situation where here's somebody essentially in the ICU, initially on ECMO and shortly after with a surgical device. How fast could we do this? Well, I mentioned back in the day that we'd used a technique from Stephen Kingsmore, who at the time was in Kansas, now in San Diego, and he had managed to do this in 50 hours. I mentioned James Priest had managed to knock off 8 hours. So in 2014, we managed to do it in 42 hours. Stephen had gone on to do it in 26 and then 19.5 hours. Shown here is him receiving the Guinness World Record for the fastest genetic diagnosis. 19.5 hours was pretty impressive. But the genetic testing turnaround for commercial assays remains long, 56 days for standard of care. If you order a very rapid one from Baylor or Rady, it still takes 7 days. And so we wondered if we could potentially go faster. And if we could go faster, could we maintain or even improve the reach in the genome and improve the accuracy? So this was a question I asked of these individuals, particularly Slack messaged John Grzinski, a very talented PhD student, now a postdoc and staff member in the lab, also a veterinarian. Sneha, who is one of our EE students, Tanner, one of our genetics students, and Kishwar actually was at the University of California, Santa Cruz. And they came together, and we brought, in fact, a whole team together around this device. This is from a company called Oxford Nanopore. They're based in Oxford in the UK. And this is a beast, this machine. This is basically an entire sequencing center that can sit on your bench top. 48 flow cells, 500,000 little nanopores, protein pores, through which DNA will fall and the current changes, and it creates a squiggle, literally a squiggle. It's called a squigglegram. You apply deep learning methods to turn those squiggles into A's, T's, G's, and C's, and more. And we thought there was an opportunity here. If we put this entire machine to work, we could potentially sequence a genome in just a few hours, maybe even 1 to 2. But then we'd have the data to deal with. So we pulled together a big team, as we had before. Oh, sorry, my dog is excited. Google Health, UC Santa Cruz, NVIDIA, who are famous, of course, for acceleration GPUs, Baylor College of Medicine and Oxford Nanopore. And we started meeting to work out whether this was even possible. And what about the steps? I showed them to you earlier. Of course, we need DNA extraction. First of all, we want to do that as fast as possible. So let's try to optimize the wet lab. Then we do the sequencing. Of course, we turn on this whole machine and see if we can get DNA off it as fast as anyone ever has. And then we have this complicated set of algorithms. And I won't go into these in a lot of detail, but suffice it to say, each one of these requires a different optimization. So these ones based around deep learning are better optimized by graphical processor units, GPUs. Other ones like the alignment that I mentioned earlier are better optimized in a linear fashion on a CPU. So a fairly complicated set of computations, then that variant prioritization, and finally, we hope, a diagnosis. So let's show you how this happened for Matthew. First of all, 8 libraries put across this entire machine. This part took a total of 4.2 hours. So we can stick that up against our world record of 19.5 hours. Then the sequencing part. Sorry, I'm going to let my dog out. Or else he will, sorry, he will never be quiet. The sequencing part, how long did that take? That took a total of 6.9 hours. So we have a genome in hand at 7 hours. This is getting more exciting. So then we have to do all that computation. Now, initially, that took 18 hours for the machine that came with the sequencer. Through very important work from Sneha, she managed to reduce that to basically just a half hour on top of the sequencing time. So it was almost happening in real time. Total of 3.6 hours, but overlapping with the sequencing, taking us to 10.5 hours, at which time we had the variant list. The variant list, unfortunately, is 4.5 million variants long. So to make a diagnosis, we have to find the one variant. But through the clinical genome program team here at Stanford, they applied the variants and found this duplication in troponin T, a gene known to cause dilated cardiomyopathy, a duplication in a repeat region after 46 minutes. So for Matthew's genome, we were able to make this diagnosis 11.25 hours through a whole genome sequencing approach after the blood was drawn. In fact, when we did this in our pilot, and then for Matthew, that he was urgently listed for transplant as a result of this confirmation that this was a genetic diagnosis. And in fact, he underwent transplant 21 days later. This is him immediately, or just I think a day after his transplant. Even better, this is him 6 months after his transplant, standing next to the machine that sequenced his genome in record time. And when the paper came out, we were able, as it turned out, for each of the patients that we did in this pilot to do it faster than the previous world record of 19.5 hours. About halfway through, we actually altered our approach a little more and made all of the last 5 or 6 diagnoses in under 10 hours total. So we had managed to move this to something that could potentially be available at the end of your shift if you send the test at the beginning. Certainly not where we were at the beginning, when the genome took 10 years, 10 countries, and $3 billion. These are the sorts of diagnoses that were possible. But you can see that this is a diverse group, both in terms of age, everything from just a few months old. In fact, we certainly looked at neonates here, one 2 weeks old, all the way up to someone in our adult ICU at 57 years. Every single one of the diagnoses that were made was immediately actionable. And this has been shown by others to be highly cost-effective in an intensive care environment. So where does it look here? Well, 56 days down to 7 days, down to 5. Here's our Stanford ultra-rapid genome, just in around 8 hours. And if we put this on our kind of Moore's Law graph, here's the Quake genome with which I started today's stories. The Human Genome Project, of course, these are the number of hours. The early Illumina genomes just before the Quake one, this was 5 days. Here's Stephen Kingsmore's world records, and then the new Stanford genomes from just last year. These are also long-read genomes, and I didn't spend a lot of time talking about that today, but there are major advantages to having long reads when we think about trying to make diagnoses, especially in newborns with potential neurodegenerative or neurodevelopmental disease. We did let Guinness know, and in fact, they did award us, as was mentioned at the beginning, a new Guinness World Record. Interestingly, we were talking to 2 different journals about this at the time, and Guinness, and in fact, to our surprise, Guinness were probably, there were more emails with Guinness than there were, let me just put it that way, with the New England Journal and Nature Biotechnology. This was the first time that the whole team came together. This is a large balloon sculpture from one of our patients of DNA, of course, and this was the Guinness World Records. You can see just how many people it takes to try to break these kind of boundaries and move forward, a really huge team effort. And this is what it looks like late at night when you realize you have likely made a diagnosis. These are genetic counselors, our clinical genome team. This is the Zoom call, I think at 10 or 11 p.m. I can't remember if this was actually Matthew's diagnosis, but they often happened late. There were also times when not only was the diagnosis part happening late, but I came into my office one day and saw this bag and found that also the sequencing had been happening overnight. In order that we could get the data ready for first thing in the morning. So genomics overnight is not something that we've really thought about in the past, but perhaps something that we need to start to think about if we're thinking about critical care. Well, we talked about how much this car had dropped in cost, and we're heading towards the 1 cent Ferrari, but also we've managed to speed it up. This car goes at 199 miles per hour. But what if we speeded up this car as much as we managed to speed up human genome sequencing? Well, it would hit 32 million miles per hour, which isn't too bad. And perhaps we need a new comparator vehicle. I was sorry to say that actually this isn't quite warp speed. So we have still some work to do before we call it good on that. These are the 3 papers. If you're interested in reading more, they just came out earlier this year. There's a lot of detail, in particular technical detail here in this Nature Biotechnology paper. The clinical work is in the New England Journal, and Matthew's case report described in a bit more detail here in the Circulation Genetics Journal. So in conclusion, I have told you some stories and given you a kind of 10-year history of the last 10 to 12 years of genome sequencing for medicine. We've seen these dramatic reductions in cost, improvements in the quality. I didn't talk too much about that today, but that's been really a major part of the work we've done in the last 10 years, just to try to improve the quality of calling of genome sequencing data. But that, I think, has paved the way for the broad application of clinical whole genome sequencing in medicine beyond just rare disease. And in particular, although rare disease is where much of the work has been done to date, I think we're about to move towards using this for preventive care through polygenic risk scores, pharmacogenomics. The applications are expansive. In fact, at Stanford today, we have a whole genome backbone, thanks to Stuart Scott and the team at the Clinical Genome Program. We have a genome backbone for much of our genetic testing, including in particular cardiovascular and renal panels, which allows us a real interest in terms of science that can be done with this, but also just much more likely to find diagnoses and account for different kinds of variants that wouldn't be found with a standard exome or panel-based approach. Finally, in critical care, when speed is required, we are able to do this faster now, and we can now make a whole genome-based diagnosis routinely in under 8 hours. And just to mention that, as Eldred mentioned at the beginning, I find these patient stories so incredibly moving and touching, and our ability to spend time with these rare disease patients in particular. I've also always been fascinated and so much enjoyed writing, of course, by Abraham and others, and thought I would try and give it a shot. With his encouragement, I did. And so if you're interested in reading these kind of stories, including most of them from patients at Stanford Hospital and rounds like the one I'm going to later this morning, then you're welcome, of course, to take a read or even a listen. They actually, believe it or not, allowed me to narrate the book, although I had to do an audition in case nobody could understand my Scottish accent. Fortunately, I passed. This work is really not mine. This work is a very, very large number of people, and I get to tell their stories, and that's a huge privilege for me. This is our lab team. A little out of date. We need a new photo. I think we'll aim for that this fall. Here are the UDN team, the Undiagnosed Diseases Center here at Stanford, led by John Bernstein and Matt Wheeler, and many other contributions from others you see in this photo. The National Undiagnosed Disease Network, too, now in transition to a new funding mechanism. The Clinical Genome Program team here working on CLIA genomes. So when you order a genome through Epic, this is the team that will deliver the result back to you. And finally, a couple of quick plugs for other programs. This is not a genome program, but I'm lucky enough to be able to co-direct the Stanford Medicine Catalyst program, which is a new funded mechanism to move ideas, not just genomes, but ideas really across all verticals, anything that could transform or impact medicine here at Stanford or beyond. And we're interested in funding programs. We have an RFA every year where we push programs forward. And we're interested in whether clinical fellows might be interested in joining us to help us look at these ideas, help move them forward. And if you are interested, please give me a call or Kevin there. Finally, also, we have a very exciting new Human Performance Alliance. My dog is very excited by it. And we have a clinician scientist fellowship program. So this is full funding for 2 years of dedicated research for a broadly defined human performance. So if any of you are heading towards your research years and are working on something that could be within this range, please think about this. The QR codes are here. Contact me if you're interested. All right, and then this was probably one of our proudest moments. In the middle of all the bad news we've had in the last 2 years, we released the work from the Undiagnosed Diseases Network, and we're featured in the week in Good News in New York, in the New York Times. And I think if we're stretching for anything in medicine, is to be able to give our patients good news, and we feel really privileged every day when we are able to do that. Thanks for joining. I would be delighted to take any questions.
Dr. Ashley, thank you for that amazing presentation. A quick call to everybody. Please do put the questions in there, and do, if they're expecting a lot of questions, vote them up. Dr. Ashley, one of the things I know through our communications team is you are one of the most prolific faculty members when we see just work coming through, all the things that you're doing. And what's amazing about this talk is it sounds like despite all the stuff you've done, it feels like you're just even getting started, and the future is just insane. I was hoping to be able to get this just general question, and you made such an amazing, just shared how things are changing so much and are going to change dramatically just by cost and speed and whatnot. Can you tell us a little bit what we expect as clinicians? But we've been hearing about precision medicine for a number of years since a lot of the work you did started going. What do we expect? Patients are going to be showing up in the hospital or in clinic with their data. Will they be able? I understand, a lot of the genome companies will start doing translating genomes, but what do they do with that data? Will they actually interpret it for you? That's a huge aspect as well that you guys do so well.
No, it's a really important point. Oh, yeah, and I think the data becomes available, but what happens next? Clearly, companies like 23andMe have been offering direct-to-consumer testing. It's not whole genomes. Importantly, that's a chip that would be the equivalent of giving you kind of the postcodes for your genome, but not every single letter. We'll be able to diagnose rare disease, but there's definitely information, particularly on ancestry and risk for different diseases that you could get through that. And we certainly have people coming to the clinic with their 23andMe data and asking us to help them analyze it. There's been a kind of rise and fall of a few companies doing direct to consumer genomes. Actually, at the moment, there isn't really a mechanism for doing that. So almost, and I think that's probably fair right now, it's sufficiently complex that you really do need a whole team of people to help analyze it. And so I think mostly this is happening within clinical medicine. I think, of course, it initially started within medical genetics, within cancer, and within cardiovascular. But really, if you think about it, every subspecialty we have has a portion of cases that are clearly Mendelian, clearly mostly genetic. And then in terms of the common diseases, every single disease we have has some degree of genetic component. It may be a very small part, but it is some component. Even obesity, for example, that we think of as a almost purely lifestyle environmental, there's clearly genetic predisposition there too. So I do think it's coming. I mentioned the polygenic risk scores, and we are beginning this fall, one of the first medical centers really anywhere to introduce an integrated score for coronary artery disease. So if you come to one of our preventive cardiology clinics or even primary care, you will be able to have a score that integrates both your genetic risk and your environmental and lifestyle risks, so nature and nurture together. And as you can see from the C statistic, it's not a huge bump, but it is definitely a more precise and better estimate of your risk on which we can base that when we have that information.
There's a question here asking, could you reach the same speed with the Illumina sequencing? You mentioned, I think Illumina is the biggest company that does sequencing right now, and they're working on, I believe, some faster and less expensive approaches as well.
They are, yeah. We've been looking to them as the leader to kind of push the price down. I mean, that Moore's Law graph, actually has kind of leveled off a little bit, partly because they haven't had much competition to push it lower. There are now actually the patent on the original method, sequencing by synthesis, it's called, actually expired earlier this year, and 3 or 4 companies have now launched with kind of upgraded versions of that. So that will provide some competitive forces there to push this down perhaps further. As you mentioned, they've not stood still. We would not expect them to, and they're a very exciting company. I think we've actually been working with them on their long read approach. So they're looking to try and get some of these advantages of long reads along with their short read machine. So we're hopefully we'll be among the earliest groups to have that available here at Stanford. On the time component, though, one of the reasons we chose to work with Nanopore is that it's actually not possible to go much faster on Illumina, partly because of the key is in that word synthesis. Basically, in order to do Illumina sequencing, you're actually synthesizing DNA, reading each fluorescent base as it's added by DNA polymerase. And there's a limit to how fast that can be. In fact, we didn't think it was faster to do it than 18 hours on a Illumina machine. But while we were doing this work, competition, of course, is fun. Stephen Kingsmore, who's a fellow Scot, by the way, he has an Irish accent, but fellow Scot, he actually pushed his own number down and published a paper in the New England Journal at 14 hours. So there's a little bit of competitive force in there as well, just trying to see who can do this faster. But he also mentioned he didn't think he could get the Illumina machine to go any faster than that. And I think that's probably true with that technology.
Dr. Ashley, can I ask while we have you as well? So you talked a good amount about the undiagnosed disease network. Many of us clinicians know that exists. Is there any sort of, and I've referred patients to it, been a while, but can you share any logistical information about how our own faculty, if they have that, when we have that rare, by the way, it's been an amazing resource to have when we have a difficult case to be able, and the stress that the patients get to be able to say, well, we have this opportunity with this undiagnosed this network at Stanford. How can you, any other information you'd like to share just logistically to make sure our clinicians can refer to it?
Yeah, well, in fact, anyone can refer with one caveat that I'll mention in a moment. But basically, even the patients can self-refer, so they can go to undiagnosed.stanford.edu and follow the instructions. There does, before the referral is really formally processed, need to be a summary from a doctor, and so it's particularly helpful if the doctor and the patient decide together to do it, and then the medical summary can come in. Then it goes to coordinating center at Harvard, assuming we're the local regional site. It comes back to us, and then we start to gather the records, and they pile obviously pretty high. That takes a bit of time. We work through them and then decide really as a national group whether to take on any individual cases. One small caveat right now, I mentioned it a couple of times, is that we're in transition. So officially right this moment, unfortunately, we're not accepting new patients. This was funded by the Common Fund, which is an NIH Directors Fund that has a limitation of 10 years. And so after 10 years, the funding has to change. A little bit unclear exactly what it looks like going forward. At the moment, there's only clear funding for a coordinating center. There isn't actually funding for the clinical sites at the moment, but we're hoping over the course of the next year or so to be able to kind of reopen again and basically move forward. Otherwise, we'll need to look at other funding mechanisms. But we encourage everyone to reach out anyway if you have a particularly tough case. We obviously have some choices to make in terms of which ones we accept, but we use a kind of rubric that if the ones we think we're most likely to be able to help are the ones that we accept. So definitely available on undiagnosed.stanford.edu is where to go for information.
Dr. Ashley, I got a question that just popped in with Lafayette. It's okay if I answer that one? We're a couple minutes past 9, and then we'll close. Dr. Lafayette, thanks so much. Dr. Lafayette asks, we are at least as excited as your fantastic progress. What of the many other findings relevant and potentially relevant that come up? How do you or an army of genetic counselors deal with this? Best to just look for an answer for a given question, or release the whole story, complete data?
Yeah, no, that's a really good question. Because of course, if you're analyzing an entire genome, you're going to find everything, including the answers to questions you didn't even ask. And that's what we did at the beginning, because we were partly trying to work out just how to deal with this data. So we tried to kind of answer every question. But in reality, I think most of the time, as Rich mentions in the question, the better answer is to take a focused question. But the good news is that now, if you have one of these tests at Stanford, your genome is now part of your medical record. And so the next time you need a genetic test, well, you don't actually need the test. We just need to go look up your genome for the next question. And of course, there may be an option in time if there are questions you like to answer that relate to maybe a risk of coronary artery disease that we mentioned, or polygenic risk score for diabetes, that can be pulled from your medical record. So we see the future as one where at some point, likely in your adult life, but some people are suggesting doing it as newborns, your genome is sequenced and then used as part of your medical care throughout your life to help you understand better your risks for different diseases. At the moment, though, I think targeted questions, targeted answers is definitely the way to deal with the variants of unknown significance.
Dr. Ashley, thank you so much. Right, 9:03 should probably close it down. And Clark, I sent the updated email. We'll send out the Undiagnosed Network from Stanford link with the follow-up from this presentation as well. I followed Homer as an author on Amazon. Thank you for the tip in case anything new comes out. Thank you so much for all the amazing work you do, Dr. Ashley. Thanks for being with us today, and thanks for everybody for sticking with us. Hope you have a great rest of the day.